There is a single ant at the base of a cecropia tree in the forests of Panama, and it is carrying a piece of leaf that weighs 50 times what it does. Not struggling, not stumbling, moving with the specific purposeful steadiness of something that has done this 10,000 times before, and will do it 10,000 times again, and has never once required anyone to tell it to do it, has never once received a schedule, or a work order, or signal from any central authority designating this task to this individual at this moment. It found the leaf, it cut the piece, it is carrying it home, and when it arrives at the colony entrance and descends into the fungus gardens 3 meters below the surface and deposits the leaf piece with the gardener ants who will process it, it will turn around and go back to the tree, not because anything told it to, because that is what the colony needs, and the colony has no mechanism for conveying what it needs except through a distributed chemical conversation that this ant is reading and contributing to simultaneously without any awareness that the conversation exists. Somewhere in the colony, which houses somewhere between 1 million and 8 million individuals, depending on the species and the age of the colony and the season, and a dozen other variables that the colony itself tracks, in aggregate, without any individual tracking any of them. A queen is laying eggs at a rate of several thousand per day. She is not giving orders. She has never given an order in her life. She does not know how many foragers are working the tree line, or whether the fungus gardens have enough material, or whether the soldiers stationed near the eastern entrance need to be reinforced. She knows none of this because she cannot know any of it. Her brain is not larger than the brain of the worker carrying the leaf piece. She is not the mind of the colony. She is its reproductive system, and the colony is governed by something for which human language does not have a clean word, something that biologists describe as emergent collective intelligence, and which is, in the full honest assessment of what it does and how long it has been doing it, more stable, more adaptive, and more consistently successful than almost any organizational system the human species has ever designed. Ant colonies have existed in recognizable form for approximately 130 million years. They were present when dinosaurs were the dominant large animals on land. They survived the extinction event that ended the Mesozoic era. They survived every subsequent climate shift, every ice age, every period of ecological reorganization that the planet has gone through since. Today, ant species constitute an estimated 15 to 25% of terrestrial animal biomass, meaning that if you weighed every land animal on Earth and set the weight of all ants against the weight of everything else, the ants would represent between 1/6 and 1/4 of the total. They occupy every terrestrial ecosystem on the planet, except the polar ice sheets. They have achieved this not through the physical power of any individual ant, which is negligible, but through the organizational principle by which individual ants aggregate into something that behaves, at the colony level, like a single organism with a distributed brain and no single point of failure, and a capacity to absorb damage and loss at a scale that would destroy any centrally organized system. Understanding how this works requires starting with the thing that most people who think about ant colonies think is the explanation, and is actually not the explanation at all. The queen does not run the colony. This is the most persistent and most consequential misunderstanding in popular accounts of ant biology, and it matters because the queen, as central controller, is such an intuitive model that it actively prevents understanding of what is actually happening. The queen is larger than the workers. She lives longer than the workers. She is the colony's only source of new workers, since in most ant species, only the queen produces viable female eggs, and the workers are all female. But her relationship to the daily operational decisions of the colony, the foraging routes, and the nest construction, and the defense responses, and the allocation of workers to tasks, is essentially zero. She does not communicate operational instructions to the colony. She does not receive operational reports from the colony. She is, from the perspective of colony management, a very large egg-laying machine around whom the workers maintain appropriate temperature and humidity and food supply. Not because she has commanded them to do so, but because maintaining the queen is a behavior pattern the workers exhibit in the presence of the chemical signals the queen's body produces. Remove the queen from an established colony of many ant species, and the colony continues functioning. The foragers continue foraging, the soldiers continue patrolling, the nurses continue tending the brood that exists in the nest at the time of removal. The colony will eventually decline and fail in most species, because without a laying queen, it cannot replace workers that die. And ant workers have lifespans measured in weeks or months, but the decline is slow and orderly. It does not resemble the collapse of a system whose central controller has been removed. It resembles the gradual reduction of a workforce that can no longer recruit new members. The colony does not become disorganized. It becomes smaller. There is a difference, and the difference is the entire story of how ant colonies actually work. The colony is governed by chemistry. Every individual ant in a colony is simultaneously transmitting and receiving chemical information encoded in compounds called pheromones, and the aggregate of all of that, transmission and reception at any given moment, constitutes something that functions as a colony-level awareness of conditions, needs, and states that no individual ant anywhere in the system possesses. An individual ant knows what its own sensors are detecting. It knows what chemical signals are present in its immediate environment. It knows in a functional sense what task it is currently performing and whether the chemical context around it is consistent with continuing that task or shifting to a different one. It does not know what is happening on the other side of the colony. It does not need to. The pheromone communication system operates through a layered architecture of signals at different temporal scales and carrying different kinds of information. Alarm pheromones are short-range and fast-degrading, designed to communicate immediate local threat and produce immediate local response without propagating false alarms to distant colony areas where the threat is not present. Recruitment pheromones are more persistent and longer range, designed to direct other workers toward locations of interest, whether a food source or a site requiring defensive attention. Trail pheromones are deposited by returning foragers along the routes they have used and are reinforced by subsequent foragers who use the same route, building up concentration along successful paths and fading from abandoned ones in a way that continuously adjusts the foraging network toward currently productive areas without any planner having designed the adjustment. The trail pheromone system produces, at the colony level, a foraging network optimization behavior that computer scientists and operations researchers have found compelling enough to model in algorithms for solving routing and network problems in human logistic systems. When foragers from a colony encounter a food source and return to the nest, they deposit trail pheromone. The more foragers that use a route, the stronger the pheromone concentration on that route, the more likely subsequent foragers are to choose it, which leads to more deposition, which leads to higher probability of choice by additional foragers. At the same time, routes that lead to exhausted or absent food sources are used less frequently. Their pheromone concentrations degrade, and they become progressively less likely to be chosen. The colony's foraging effort self-organizes toward current productive areas and away from unproductive ones, through a feedback system that requires no central monitoring, no reporting structure, and no top-down adjustment of worker assignments. The same feedback logic governs the allocation of workers to tasks across the full range of colony activities, and it is here that the colony's resilience to disruption becomes most clearly visible. An ant colony, at any given moment, has workers performing a range of different tasks: foraging, nest construction and repair, brood care, defense, waste management, food processing, queen attendance. The proportion of workers engaged in each task is not fixed by any scheduling system. It is continuously determined by the balance between chemical signals associated with each task and the current pool of workers whose internal state makes them receptive to those signals. When a task becomes more urgently needed, the chemical signals associated with it intensify. When a task's needs are met, its chemical signals diminish. Workers shift their task engagement in response to the chemical environment around them, and the aggregate result of all those individual shifts is a colony-level reallocation of workforce toward current needs and away from currently satisfied ones. This means that when the colony sustains losses in any particular functional group, the reallocation mechanism automatically recruits from the workers available, lose a significant fraction of your foragers to a predator attack, and the chemical signal associated with foraging need intensifies in the nest environment. Workers previously engaged in other tasks shift toward foraging. The colony absorbs the loss not by reassigning workers from a central roster, but by changing the chemical environment in a way that the workers respond to as individuals. each acting on its own local chemical context, collectively producing the reallocation that the colony-level situation requires. The flexibility of individual workers to shift tasks is itself more nuanced than the fixed caste model that most introductory accounts describe. Age polyethism is the baseline pattern in most ant species. Workers perform different tasks at different life stages, with young adults typically engaged in interior nest tasks like brood care and food processing, and older adults progressively shifting toward more dangerous exterior tasks like foraging and defense. The pattern makes adaptive sense because it exposes the youngest, most reproductively valuable workers to lower risk and reserves the higher risk exterior work for older individuals who have already made their primary contribution to colony growth. But this age-based pattern is a tendency, not a rigid assignment, and colonies regularly deviate from it when circumstances require. Experimental removals of all forager-aged workers from colonies have produced within-hours responses in which interior workers dramatically younger than the typical forager age begin performing foraging tasks. Colonies recovering from battles that have depleted their soldier population show shifts in worker behavior and even in developmental trajectories of immature workers, with more individuals developing into the soldier caste than would have under undisturbed conditions. The colony's workforce composition responds to current needs in ways that operate across timescales from hours to weeks to developmental trajectories spanning months. And at each timescale, the mechanism is not central planning but distributed response to locally detectable chemical information. The colony also manages its own health in ways that have no analog in any human organizational structure, and that researchers have only recently begun to characterize in full detail. Social immunity is the term that has emerged in the scientific literature to describe the collective disease management behaviors of social insect colonies, and the sophistication of what that phrase covers is not fully captured by either word in the combination. Individual animals have immune systems, biological mechanisms that detect and neutralize pathogens before they can cause harmful levels of damage. Social insect colonies have, in addition to the individual immune systems of their members, a set of colony-level behaviors that serve immune functions at the group level that prevent pathogens from establishing in the colony population with the same logic that public health infrastructure prevents pathogens from establishing in human populations, but implemented through behavioral mechanisms that require no central coordination and no explicit awareness of disease at any individual level. The first layer of social immunity is grooming. Ants groom each other constantly, a behavior that serves multiple functions, including the transmission of chemical information through cuticle contact, but that also removes pathogenic spores and cells from the body surfaced before they can penetrate the cuticle and cause infection. In colonies exposed to fungal pathogens, the frequency and intensity of grooming increases measurably, driven by the detection of pathogen-associated chemical compounds that trigger heightened grooming behavior in ants who contact them. The behavior is not consciously protective. The ant, detecting the foreign chemical compounds associated with fungal spores on a nestmate's cuticle, does not know what a fungal pathogen is. It responds to the chemical signal with increased grooming, and the result at the colony level is accelerated removal of pathogenic material from colony members before infection establishes. The second layer is spatial. Researchers studying how ant colonies respond to pathogen exposure have found that colonies spontaneously restructure their social networks when pathogen is detected in the population. Foragers, who have the highest exposure to environmental pathogens and are the most likely to bring novel pathogens into the nest, are socially separated from the queen and brood in ways that reduce the probability that a pathogen introduced by a forager reaches the colony's most reproductively critical members. This separation is not organized by any individual who understands the epidemiological logic. It emerges from changes in the chemical environment associated with forager activity that alter how other workers interact with foragers, producing a network restructuring that has the effect of epidemic compartmentalization without any individual having planned for it. The third layer is the undertaker system.
Every ant colony produces a subset of workers whose task is the removal of dead colony members from the nest. Dead ants produce oleic acid and other chemical compounds associated with decomposition, and workers detecting those compounds respond with removal behavior, carrying the dead individual to a refuse area outside the nest, regardless of whether they are designated undertakers or not. The specificity of the chemical trigger means that healthy ants are not removed while showing any signs of distress, but dead ants are removed quickly and efficiently from the nest environment before the pathogens they may be carrying have time to spread to living colony members.
In colonies facing active pathogen challenge, the undertaker system is supplemented by another behavior. Workers who have been heavily exposed to pathogens voluntarily leave the nest and do not return. This self-removal behavior, documented in multiple ant species exposed to entomopathogenic fungi, has the colony-level effect of removing the most contaminated individuals from the shared nest environment before those individuals die and become pathogen sources inside the nest. The behavior is not suicide in any motivated sense. It is the behavioral consequence of a heavily contaminated ant's chemical environment changing in ways that produce exit behavior rather than nest-oriented behavior, and the colony-level result is the removal of the highest-risk individuals from the protected interior. Together, these layers of social immunity produce a colony-level disease management capacity that researchers have described as exceeding what would be predicted from the individual immune systems of the colony members alone. The colony is healthier than its members would be if they lived individually. The thing that makes all of this possible, the architecture that allows individual ant behavior to aggregate into colony-level adaptive intelligence, is something that researchers studying complex systems have named stigmergy and have found operating in ant colonies in a purer and more legible form than almost anywhere else in biology. Stigmergy is indirect coordination through the environment. Rather than communicating directly with each other to coordinate activity, individuals modify the environment in ways that influence the behavior of other individuals who encounter those modifications, who then modify the environment further, producing organized, collective behavior without any direct communication between the individuals involved or any central entity coordinating the whole. The pheromone trail is the canonical example. The forager that finds food does not contact other foragers and explain the food location. It deposits a chemical modification of the environment on the route between the food and the nest. Other foragers who encounter that chemical modification respond to it in predictable ways, depositing their own modifications if they use the route, producing a self-organizing information infrastructure about food location that exists in the physical environment, is readable by any worker who contacts it, and requires no direct communication between any two individuals to maintain. Stigmergy is also the mechanism behind ant nest architecture, which is among the most complex self-organized physical structures produced by any animal species, and which is produced without any ant having a plan of the finished structure, without any ant coordinating the placement of other ants' contributions, and without any ant knowing what the full structure looks like at any point during construction. Each worker deposits material, moves material, or removes material based on what it detects in its immediate local environment. The temperature, humidity, chemical composition, existing structural geometry of the surface it is working on. The result of hundreds of thousands of individual workers, each making local decisions based on local information, is a structure with humidity-regulated chamber systems, ventilation architecture that maintains gas concentrations appropriate for the colony, a spatial organization of functions across depth levels that reflects the thermal and humidity gradients of the soil, and an overall structural integrity that engineering analysis has found to be highly optimized for the mechanical properties of the material and the loads the structure must support. No ant designed this. No ant could design this. The design is an emergent property of the collective behavior of workers, each responding to their local environment, and the local environments are themselves shaped by the collective behavior of all the workers, producing a feedback system that converges on adaptive structural solutions through a process that is functionally equivalent to optimization without any individual having performed an optimization. Flood a section of the nest. The workers in the flooded section move upward. They begin rebuilding. The workers elsewhere in the nest do not receive a crisis communication from a central authority. They detect the changes in the chemical environment associated with the disturbed nest section, and they respond to those changes with repair behavior. The nest rebuilds from every point at which repair behavior is triggered, simultaneously, without any worker knowing what the finished repaired nest will look like, without any worker knowing what other workers are doing in other parts of the nest. The nest rebuilds anyway. Destroy a major foraging trail. The pheromone signal on that trail degrades when foragers stop reinforcing it. Workers begin scouting in new directions. When a successful new route is established, pheromone deposition begins. The trail strengthens. Foraging shifts to the new route. The colony loses foraging efficiency during the transition, and then regains it, without any worker having understood the problem or designed the solution. Remove 50% of the worker population. This has been done experimentally with established colonies, and the result's not colony collapse. The result is colony contraction followed by recovery. The survivors continue all essential functions. The queen continues laying. The brood continues developing. The remaining workers collectively shift their task engagement toward the most critical functions. Over weeks and months, as the new workers from surviving brood mature, the population rebuilds. The colony that lost 50% of its workers does not behave like an organization that has lost 50% of its staff. It behaves like an organism that has healed from a significant injury. To understand what 130 million years of continuous operation actually means, it helps to place it against the timeline of things humans tend to think of as ancient. The Roman Empire controlled territory for roughly 500 years before its western portion collapsed. Egyptian civilization sustained cultural and political continuity for approximately 3,000 years. Agriculture is approximately 12,000 years old. Writing is approximately 5,000 years old. The oldest surviving human institution measured by continuous, uninterrupted operation is measured in centuries. Ant colonies have been running the same organizational model for 130 million years. The Cretaceous-Paleogene extinction event 66 million years ago, which ended the Mesozoic era and eliminated approximately 3/4 of all species on Earth, including the non-avian dinosaurs, did not eliminate ant colonies. The fossil record shows ant species on both sides of that boundary, reduced in diversity in the immediate aftermath, and then diversifying again as ecological niches vacated by the extinction were filled by new species. Whatever conditions that event produced, whatever cascading ecological changes followed from the initial impact in the years of altered atmosphere and reduced photosynthesis, the distributed colonial organization was flexible enough and robust enough that ant lineages pulled through it, while the majority of animal life on the planet did not. The question of what would actually collapse a well-established ant colony, as opposed to simply reducing its population or limiting its foraging territory, is one that researchers find genuinely difficult to answer, because the answer is almost nothing that the natural environment reliably produces. Catastrophic nest destruction forces relocation, not collapse. Severe population loss produces contraction and recovery rather than failure. Disease exposure produces the social immune response. Environmental stress produces behavioral and physiological plasticity that allows colonies to maintain function across temperature ranges, moisture ranges, and food availability ranges that would be lethal to most individual animals. The primary mechanism by which established ant colonies are actually destroyed is other ants, in territory conflicts that deplete worker populations below the threshold at which essential functions can be maintained. And even then, the process is slow enough that it resembles attrition more than collapse. Ant colony optimization algorithms, computational procedures modeled on the pheromone trail reinforcement system, are today used in logistics, telecommunications network routing, and scheduling optimization, producing solutions to combinatorial problems that conventional search algorithms find computationally intractable. The collective intelligence of the ant colony has been formalized mathematically and implemented in computer systems that solve real engineering problems, competing with or outperforming solutions generated by humans who understand the problem completely and are deliberately trying to optimize it. The ant cutting its leaf piece at the base of the Cecropia tree does not know any of this. It does not know it is part of a colony. It does not know the colony exists as a level of organization above itself. It knows what it can detect through its antennae and its body surface in the few centimeters of space around it, and it responds to that detection in the way its nervous system has been shaped by 130 million years of selection to respond. And from that, the colony that has survived everything the world has produced across 130 million years continues to function exactly as it needs to, without anyone in charge, without any single point of failure, without any mechanism for catastrophic collapse that the natural environment has ever reliably been able to produce. The leaf piece is cut. The ant turns toward home. It will be back. No one told it to. No one ever has.